I will go ahead and get started. Good afternoon, everyone. Thank you very much for joining us on our fourth Facts and Snacks Lunchtime Journal Club discussion, where we discuss COVID-19 related research and how it affects our communities. My name is Elisha Barrientos, your host and a community engagement specialist at the Research Centers in Minority Institutions, or RCMI, Community Engagement Core, led by Dr. Carla Williams. Our group's primary goal is to ensure that RCMI-supported research addresses the needs and interests of the local community. To achieve this, we partner with local organizations to address health-related concerns, promote community participation in research and partnership with researchers in study design and execution, and ensure research findings are shared with relevant communities. The CEC offers consultations to Howard research teams to incorporate community engagement best practices into their projects, as well as facilitate community partnership, introductions, and more. I am pleased to welcome Dr. Thomas Heinbockel as our speaker this afternoon. Dr. Heinbockel is currently professor in the Department of Anatomy at Howard University College of Medicine. Dr. Heinbockel's laboratory engages in multidisciplinary research to elucidate organizational principles of neural systems in the brain, specifically the limbic and olfactory system. His research has been directed at understanding brain mechanisms of information, processing, and their relation to neurological and neuropsychiatric disorders. Dr. Heinbockel studied biology at the Philipps University, Marburg, Germany. His studies of the brain started during his master's thesis work at the Max Planck Institute for Behavioral Physiology, Starnberg-Seewiesen, Germany. Subsequently, he completed a PhD in neuroscience at the University of Arizona, Tucson, Arizona. After graduating, he was research associate at the Institute of Physiology, Otto von Guericke University School of Medicine at Magdeburg, Germany. Prior to his arrival at Howard University, Dr. Heinbockel held joint research faculty appointments in the Department of Anatomy and Neurobiology and the Department of Physiology at the University of Maryland School of Medicine in Baltimore, Maryland. He still maintains an adjunct appointment in these departments. Thank you so much for joining us today, Dr. Heinbockel. Please feel free to share your screen.
Great. Thank you very much for the kind introduction. Very nice. And good afternoon, everyone. It's my pleasure to be here, and I'm honored to be asked to present here during this Journal Club. And so the topic of today's Journal Club is really based on two review articles that I published not too long ago, and they really deal with COVID-induced Anosmia-Ageusia recovery using phytochemicals and essential oils. And so I've been involved in COVID research for basically ever since it started, and I will tell you a little more about that. And as you all know, COVID has led in quite a few of the patients to a loss of smell and taste as symptoms of the disease, and that is true. So for quite a few patients, in the sense that the loss of smell was actually considered to be a distinctive feature of the disease, and sort of a indication that people actually have COVID. If someone all of a sudden had a loss of smell, then that was considered to be a good indicator that someone was infected with COVID. And so it turns out that quite a few, almost everyone, had some level of smell dysfunction, and also quite a few people had taste dysfunctions affecting one or several of our taste sensations or taste qualities: salty, sour, bitter, umami, and so on. About half of the patients showed dysfunction in 2 or more of these taste qualities. 11% reported impairment in a single taste quality, and salty taste, and we're going to come back to that very late in the talk, was most frequently affected. And then there's this other sense. So when we talk about our chemical senses, we typically talk about smell and taste, but there's also this chemisthesis, especially our ability to detect something hot and spicy in the mouth, and that was also often affected. So basically both or all three, smell, taste, and chemisthesis, were affected by COVID. And so there's a lot of terms, and you don't need to remember all of them. I just want to have a slide that actually mentions them, because some of the terms I might come back to. Anosmia is a complete loss of smell. Then there's hyposmia, a weakened sense of smell. Parosmia, distorted. Phantosmia, people smell something that isn't even there. Or dysosmia is just a general dysfunction. When we talk about smell, typically we talk about smell coming right into the nose from the front of the nose. That's also referred to as orthonasal olfaction. But then there's also this retronasal olfaction, which basically means you're eating something and there's odors emanating from the food, and they basically go through the back of your mouth, and then they reach the olfactory epithelium. So that would be retronasal olfaction. And that becomes important because that leads us to another term that's known as flavor. It's basically the sense of smell that comes from odorants in foods and drinks, that are really in the oral cavity, and then the odorants travel to the olfactory epithelium, to the nose or to the nasal epithelium, where they are then detected. Again, this is the retronasal odor perception. So now similar terms apply to the loss of taste, ageusia, the loss of sense of taste, hypo, weakened, less sense of taste actually shouldn't be smell. Norm is normal, distorted is distorted sense of taste. Phantogeusia, people taste something that isn't even there, or they just have a dysfunction. And then again, chemisthesis is a sense of hot and spicy items, food items, mediated by this relatively new class of receptor channels, the TRP channels, transient receptor potential channels, and there's a couple of different types of them that are activated by different types of molecules. So when we talk about taste and smell, and again, when we talk about taste in particular, taste or really is flavor is 80% smell through this retronasal activation, because on the tongue, we have, in addition to taste receptors, we have also thermoreceptors, mechanoreceptors, pain receptors, and all of that influences taste, and it can either enhance or detract from the taste. But taste is really in the mouth. It's not in the nose, because we have taste receptor cells in the tongue or on the tongue. But because we have this coincidence of somatosensory stimulation through all these receptors, and then we have that retronasal passage of odorants into the nose, that's why we often refer to that as flavor. So odorants are perceived as flavor in the mouth. And that's why when people say, oh, I have a cold, I can't taste my food anymore, what they're really talking about is this lack of retronasal passage of the odorants into the nose. So again, here the chemical senses, and I just focus on these two here, taste and olfaction, or gustation and olfaction. And so basically any tastings or even the odorants that come into the nose, they must be dissolved in an aqueous solution. And in the mouth, that's immediately obvious. That's dissolved in saliva. And again, we're going to come back to that at the very end. Smell, also the odorant molecules need to be dissolved in fluids of the nasal membranes. Now, the olfactory system typically deals with general odors, volatiles, pheromones, food odors, predator odors, mating odors, aggression cues, and the odor signals convey individual identity, whereas the gustatory system typically deals with the non-volatiles. And we have these different taste sensations or different taste qualities, sweet, bitter, salty, sour, and umami, the more recent one. Now, what do the chemical senses really need to do? Well, they're almost like the immune system. They need to detect all relevant chemicals in the environment, including novel ones. And then the chemical senses need to encode the quality and the quantity of the individual chemicals in the context of complex chemical mixtures, because typically when we smell or taste something, we're not just smelling or tasting an individual molecule or chemical. We are really sensing a complex chemical mixtures. And so we need to be able to decode this and interpret this information in the context of what we already know or in the context of other sensory input. Again, that could be that somatosensory input. And then our nervous system generates an appropriate behavioral response. So now, having told you all of this, I should point out that like most of us, we haven't dealt with coronaviruses or COVID, obviously, not before the pandemic. And really, as was pointed out in the introduction, this is sort of really where I'm coming from, talking about systems, cellular systems, neuroscience. And I'm typically dealing with, questions such as what is the structural and functional organization of the olfactory and limbic system, and what are the neuromechanisms underlying signaling in these systems, and then how is sensory or synaptic information processed and integrated in the brain. And if you want to put that into an even larger context, sort of a translational medicine context, then my research is really there to expedite the discovery of new diagnostic tools and treatments using a multidisciplinary, highly collaborative bench-to-bedside approach. And it's really driven by patients and their diseases, which is sort of guiding our primary focus. And then the research spans everywhere from molecular cellular biology to preclinical models and essentially back again. And along the way, we train the next generation of researchers to be effective translators of biomedical discovery. And so we are operating at the intersection of clinical and related sciences, and hope to have the expertise to generate and lead discovery through an integrated process, and by doing so, increasing the efficiency of translating science knowledge into health improvements. So again, my journey into COVID research started pretty much when COVID hit the country in March of 2020. And initially there were a number of these anecdotal reports that people who had COVID lost their sense of smell and taste, namely those that tested positive for COVID. And so based on that, a group of international smell and taste researchers got together and they said, look, we need to look into this because obviously it's affecting the chemical senses. We want to know what's going on. We want to learn about the coronavirus. So a new group was founded, the Global Consortium for Chemosensory Research, has about 600 members. They're from all over the world, and we are conducting surveys with thousands of people that respond to questionnaires, including service on the recovery process now. And there are a number of ongoing projects. And so I'm part of this group, and that sort of led me into looking more into what COVID-related smell and taste loss is all about. So when we talk about olfaction, let me just give you a little bit of background on what the olfactory system looks like. So principally, we are looking at air that's filled with odorants that come into the nose, and they stimulate these olfactory receptor neurons, or they basically stimulate receptor proteins here in the cilia of these olfactory receptor neurons. And these receptor neurons, they then project into the brain through this cribriform plate of the ethmoid bone. And then here in the olfactory, the so-called olfactory bulb, there's some processing going on. And then from the olfactory bulb, information is sent to higher order olfactory centers. And so if you take a closer look at the olfactory epithelium, these receptor neurons, here you see an electron micrograph showing different types of cells in the olfactory epithelium high up in the nose. Turns out that these olfactory receptor neurons, they have a fairly short lifespan. They live only for a month or two, and then they're replaced by new cells. So here you actually see one of those olfactory receptor neurons. You see the cilia coming out of this knob-like structure. So those are the receptor neurons. Then there's a basal stem cell population. They basically migrate and replace the receptor neurons, and then the receptor neurons, they form new projections as they send their axons or their processes to the olfactory bulb. And then there's another cell here that has sort of become has taken center stage in COVID. Those are support cells, also known as sustentacular cells. And the idea is that COVID does actually not affect directly, at least that's the hypothesis right now, does not directly affect the olfactory receptor neurons, but it's affecting the sustentacular or the support cells here. And by doing so, then it essentially messes up the olfactory epithelium, and that then leads to a loss of smell. So now when we talk about COVID, there's no way about getting around ACE2 or the angiotensin converting enzyme, because SARS-CoV-2, the virus that causes that causes COVID-19, that attaches to a cellular receptor, namely this angiotensin converting enzyme, ACE2, that you find here in the membrane. And so basically the virus attaches to ACE2, which is the first step that is necessary for cell entry and infection. And then the second step is that the viral and the cellular membranes actually fuse. So this ACE2 is a typical transmembrane protein, and it's found almost throughout the body on epithelial cells, endothelial cells, in the respiratory tract, heart, vasculature, kidney, parts of the GI tract. It's an ectoenzyme, meaning it actually acts outside of the cell. And the fact that it's found pretty much everywhere is probably a reason why many different organs can be affected by SARS-CoV-2. So going back to the olfactory system, if we now actually take a look at where do we find ACE2 positive cells, well, as I said, in the respiratory epithelium, in the nasal respiratory epithelium and further down, and here are sort of different cells of the respiratory epithelium, and here in that underlined in gray are the cells that are ACE2 positive. And so we find quite a few cells in the respiratory epithelium that are ACE2 positive. Same thing in the olfactory epithelium. Again, there's a couple of cells, here the basal cells, here these support cells, and then there are the receptor cells, the receptor neurons. And the receptor neurons actually do not have ACE2, suggesting again that it's not the neurons themselves that are affected directly, even though that's still a possibility, but it's an indirect effect. Now, what do we see in the taste system? Well, here's the tongue. And as I said, taste takes place on the tongue, not in the nose. On the tongue, we have about 10,000 or so taste buds. And they found in these structures known as papillae. And there's different types of papillae, filiform papillae, fungiform papillae, circumvallate papillae are these big ones here, and then foliate. And their name is essentially derived from how they look like. And so not all of them contain taste buds, but several of them contain taste buds in various quantities. And this is essentially what a taste bud looks like. And again, in the taste bud, we have basal cells that act like stem cells, and we have support cells, and then we also have receptor cells. Now, in the taste bud, it's slightly different because the receptor cells are actually not nerve cells. They're epithelial or epithelial derived cells, as opposed to the receptor neurons in the nose, which are true bipolar neurons. So again, here's a higher power shot of a taste bud. So we have supporting cells, basal cells, gustatory receptor cells, non-neural epithelial cells, and again these taste receptor cells, they don't live very long. They only live for about 10 days. So there's a very high turnover rate, both in the olfactory and in the gustatory system, which I think is probably a good thing in a way, especially if your receptor neurons are damaged somehow, then there's a relatively rapid turnover. So again, we have, if you look at the olfactory epithelium up high in the nose, these receptor neurons send their axons, their processes as cranial nerve number 1 into the brain, to the olfactory bulb. And then in the taste system, we have basically the facial nerve and the glossopharyngeal nerve that carry taste information, send it to the solitary nucleus of the medulla oblongata, and then the vagus nerve here carrying away chemocytes information, the spicy, the trigeminal nerve carries away the chemocytes information, if it's spicy, pungent, or cooling information. All of these cranial nerves are involved in some shape or form in sensing what's going on in the oral cavity. Now, again, here, the possible mechanisms of what can happen. Again, it was shown pretty early on that in COVID, obstruction is not really an issue. So it's not that the nasal mucosa is swelling up and therefore we can't swell anymore. That isn't really the case. But again, we can have this sustentacular, these supporting cell infections in the nose through SARS-CoV-2, and that leads to an ion imbalance and eventually leads to olfactory sensory neuron or olfactory receptor neuron death. There's also a case been made for these inflammatory cytokines. So there's indirect inhibition of the olfactory sensory neuron function through some intermediating cell types. And potentially, again, there's still this possibility that there's a direct infection of the olfactory sensory neurons. In the olfactory bulb, people have shown that, again, if here's some blood vessels, these are these pericytes, these are astrocytes, these are the neurons in the olfactory bulb. Apparently, SARS-CoV-2 is found in these so-called pericytes around the blood vessels, and that then this neurovascular inflammation, so that's a little blood vessel here, that can lead to damage of the olfactory bulb or the neuronal processing. For taste and kinesthesis, again, there could either be a direct infection or there's cytokines that lead to that are a result of inflammation. So essentially an indirect effect maybe on the nerves themselves rather than on the taste buds. So there's a number of possibilities, but inflammation and infection of cells are sort of key components of that. So again, here's sort of a summary slide of that. We don't know exactly what mechanism is in place. Inflammation could be in place at the olfactory membranes. So we have these pro-inflammatory cytokines, and they would basically weaken the signaling pathway from the nose to the olfactory bulb. We can have damage in the olfactory membrane, namely in the epithelium. We can have damage in the olfactory bulbs in the brain, or we could have inflammation in the brain itself. Depending on the person, multiple causes could be at work. Also, the level of damage can be different. And if there's chronic inflammation, then that might also lead to long-term problems. And we come back to that later as well. Now, I threw this slide in here because it's not that COVID-19 was the first time that people realized there's olfactory dysfunction. This is a slide that's actually quite old, but there's a number of reasons why people have issues with their sense of smell. Could be either head trauma, someone is in a car accident, or someone gets hit on the nose. Could be toxic exposure, upper respiratory tract infections, nasal sinus diseases. So there's a number of reasons why people have olfactory disorders long before COVID-19 and also long after COVID-19. But because COVID-19 hits us now, all of a sudden we realize, oh, you lose your sense of smell and taste. Well, that can have some immediate problems that you notice quickly, or others you notice more gradually. You cannot sense toxic smells, you cannot sense rotten smells, you cannot enjoy the flavor of food. It's difficult to cook because you cannot taste the food, you cannot detect smoke and fire. And then people, especially now with COVID, have found that patients with a loss of their chemical senses can fall into depression, or just experience a reduction of the quality of life, or just a reduction of their wider well-being. They might undergo mood changes. They might even have cognitive problems and develop dementia. So there's a number of reasons or a number of symptoms that might show up as a result of of the loss of sense of smell and taste. Now, when we talk about taste is defined essentially through these 5 basic taste sensations. So, and they are sweet, salty, sour, bitter, and umami. Umami, again, is this more recent one elicited by the amino acid glutamate. Sweet is obvious. Salt is evoked by metal ions, sour by hydrogen ions. Bitter is sort of like you drink that Schweppes. You drink some alkaloids in there with quinine and nicotine in it. And when you ask people, or people that have done these surveys or saw patients in the clinic, the effects of COVID was often people said, well, everything tastes kind of salty, or everything tastes bitter, or everything tastes metallic. Generally, males reported lower rates of taste loss than do females. Taste loss is highest in the middle age group, sort of 40, 50. And what often happens is that people don't really detect sweet, sour, and bitter anymore. So the threshold to detect that is higher in COVID-19 patients that have taste orders compared to patients that don't have taste orders. And there's a study that found that there's hypersensitivity to the salty quality or salty taste sensations, meaning everything tastes salty. You eat something, everything's salty. So patients show this inverse relationship to the salt threshold compared to sweet, sour, and bitter. So those are the effects. And the question, which is sort of the topic of today's Journal Club after this longer introduction, is, well, will the chemical senses come back? Is there any way to enhance recovery? And so, what are we really looking at? Well, turns out that recovery does take place. So some people recover their chemical senses quickly if they lost them, if they even lost them to begin with, maybe within 2 weeks, but many take months and still are not even really recovering. But it turns out that folks that or patients that don't have a lot of other symptoms, so the earliest recoverers, they don't have, about half of them don't have any other symptoms. Some have symptoms, half of them have other symptoms, others have not. The folks that take a longer time to recover typically have other symptoms as well. So it's not just that they don't smell or taste, but they also have some other symptoms, maybe some respiratory effects or any of the other symptoms that are associated with COVID-19. So recovery for taste seems to be better than olfactory recovery. And in patients with persistent smell loss, there are actually more inflammatory markers and positive virus RNA level in the olfactory epithelium. So the folks that, even though COVID can't really be detected anymore, they still have or it's not obvious, but you can still detect higher RNA levels in the olfactory epithelium, and those are the folks that have long-lasting loss of smell. So now, when we talk about recovery from chemosensory loss related to COVID-19, obviously there's an increase in the number of patients with post-viral olfactory dysfunction following the outbreak of COVID-19, and that just generally has raised the interest and concern about olfactory dysfunction. And unfortunately, at the present time, there's really no clear method of treatment. There's no clear treatment for this post-viral olfactory dysfunction. The best known method that people use is to improve the symptoms of olfactory dysfunction using so-called olfactory training, and they do so using essential oils. And the essential oils that are used in olfactory training typically are rose, lemon, clove, and eucalyptus. So, things that smell good or taste good. So these essential oils are selected based on some odor prism hypothesis that was proposed more than 100 years ago by a fellow by the name of Hans Henning. And so, and the term olfactory training has been used based on the concept of training olfactory sensory neurons to relearn and distinguish olfactory stimuli. At least that's sort of the idea behind it. So let me briefly go over this odor prism idea. And so when we talk about taste, let me just back up. When we talk about taste, we have these clear taste qualities or clear taste sensations that everyone agrees to, right? Sweet, salty, bitter, sour, umami. Now, Henning said, well, he said, well, I can also classify odors in a similar way. I can come up with these 6 primary categories or dimensions of how to classify odor. And he suggested that any olfactory stimulus fits into his smell prism, this 3-dimensional space. And it's based on the assumption that all odors can be analyzed into 6 primary qualities. And he basically puts these qualities at the corner of his prism. And so there's flowery, for example, violet, fruity, lemon, spicy, cloves, resinous, so pine smells, pine cones, foul, bad fish, and burnt, so something that smells burnt like tar. And so Henning postulated that if these basics are mixed, any of these are mixed, then in specific proportions, then we can essentially produce any odor that we experience. So he drew this prism and basically put these 6 primary odors at the corner. So again, here it is. People use sort of different terms here at the top, either fruity or the fruity or ethereal or fragrant or flowery. So basically, he puts these categories at the corner of his prism, and then the surfaces represent the position of odors that are similar to the primary ones that we find here at the corners. And so again, we have fragrant or flowery, like lavender, rose petals, violet, and we have ethereal or fruity, ether, like ether or cleaning fluid, or also lemon, resinous, like raisin, turpentine, or pine, and spicy, like cinnamon, nutmeg, cloves, putrid, rotten, or foul, that's essentially back here, could be like feces, rotten eggs, bad fish, and then there's burnt, like tar oil or so. So that's basically what this odor prism is supposed to show. And the reason I talk about that is because when we talk about olfactory training, a lot of the olfactory training is based on that odor prism, and a paper that was published 13 years ago by another group led by Thomas Hummel. And so he was interested, he's working at a smell clinic, and he gets all these patients that have smell dysfunctions. And so what do we do? Well, he's trying to implement this olfactory training to facilitate the recovery of the olfactory sense in patients with olfactory dysfunction. And, olfactory training is now much better known than in the pre-pandemic years because we have a lot of people who have olfactory dysfunction. And so as the name implies, the concept of olfactory training is to train the olfactory sensory neurons or the olfactory receptor neurons in the nose. So, we relearn what a rose smells like. And that's essentially what we're trying to do, relearn to smell after people were inflicted with COVID-19. And this olfactory training uses essential oils, namely rose, lemon, clove, and eucalyptus. And that's what a lot of people use. And it's based on this study by Thomas Hummel. And Hummel's study, in turn, is grounded on this odor prism hypothesis that was proposed by Henning in 1916. So for this reason, people use rose, which represents flowery, lemon representing fruity, cloves representing spicy or aromatic, and eucalyptus representing resinous. And so those are the essential oils that are used in olfactory training. People don't use rotten odors, or they don't use anything that smells burned. It's essentially sort of more pleasant odor, if you want to call it that. And so now here on this slide, you see the odor prism represented. And if you look at the bottom at the source, you will see Amazon, right? And when you see Amazon, then you know that someone's trying to make money of this. And that's exactly what happens. So on Amazon, you can find the description of the odor prism. The scientifically known odor prism classifies 4 of the main categories that make up our sense of smell. And again, we have flowery, fruity, spicy, resinous. And not surprisingly, people have come up with this idea, oh, we can market that, we can actually use that. And so here are just 3 different companies that use exactly what we described. Here's Rose, a hard time reading this, but basically they go through here, it's easier to read. Lemon, clove, rose, eucalyptus, right? So people, and here clove, lemon, eucalyptus, rose. So people are using these 4 essential oils to train their sense of smell, and it's commercially available. So essentially everyone can do that. So what did Hummel and his colleagues do? Well, so he had these participants, patients, come into the clinic. They had olfactory dysfunctions, and he gave them these 4 types of odorants, and they had to smell it in the morning and the evening, about 10 seconds each time. And they did that for 12 weeks. And he actually didn't use the entire essential oils. Instead, he just used individual odorants, but again, based on the odor prism. So he used phenyl ethyl alcohol that represented the smell of rose, representing eucalyptus, citronellal representing lemon, and eugenol representing cloves. And so he tested them before and after the 12-week period using these sniffing sticks. So you basically drop something and you sniff it on a little stick. And he tested odor discrimination, odor identification, and odor threshold. Turns out that age did not really affect the results. So you had people anywhere from 18 to 80. Age didn't really affect it very much. But what he saw was the odor threshold actually improved for essentially rose and lemon and eucalyptus. Sorry, not eucalyptus. For rose, citronellal, and eugenol, but not eucalyptus. And what he also pointed out that there was an improvement in the odor threshold. So basically people were able to smell things again, but that didn't mean that they were actually able to discriminate between the different odorants or identify specific odorants. So that suggests that sensing smell is different from recognizing the identity of an odor. Or maybe it just takes more training, and we just have grown to be able to identify different smells. But initially, he just had positive results as far as the odor threshold is concerned, not for these other two things. Now, there's a number of other people who did the same thing using the same method that Hummel used. And again, here's the odor prism. And they basically all concluded, yeah, olfactory training, that's a safe method to improve olfactory function. Higher odor concentration is better. If you use more odor types, that doesn't necessarily improve the results. And even people that have lost their sense or just have a reduced sense of smell, they show improvement. It doesn't really matter how badly your sense of smell is reduced. Olfactory training is a good thing in a way. And so other people then said, well, maybe you can actually use it together with some oral corticosteroids. That seems to improve the sense of smell significantly after loss due to COVID-19. And so again, this olfactory training has been described as kind of learning or re-educating the olfactory sensory neurons or the brain. The question, of course, is, well, is that really true? I mean, what's really happening in olfactory training? Are we really, as I said earlier, there's a high turnover of these receptor neurons, both on the tongue and in the nose? Is it just that we get a lot of, that there's just turnover and the new neurons learn something, or what's really going on? Well, there are a number of possibilities that might involve in the mechanisms that underlie olfactory training that helps us in the recovery of our olfactory sense. And one such possibility is that in these essential oils, there are chemical constituents that have some bioactive properties. And it's really these chemical constituents with these bioactive properties that help with the recovery. And maybe what they actually do is they suppress inflammation, or maybe they help with the regeneration of the nerve cells. So specifically then, people have now turned to essential oils that contain chemical constituents that have anti-inflammatory effects and that have binding affinity for SARS-CoV-2. And those could really be good candidates to test the efficiency in facilitating recovery or just helping with the recovery process. So phytochemicals are basically those chemicals that have chemicals or are chemicals that have plant origin and they have bioactive properties. And that, again, that's not new. I mean, the essential oils contain several hundreds of chemical constituents. Many of them have bioactive properties. That's not really new. And people have tried them and found them to be positive for brain function. For example, rosemary is supposed to improve memory, specifically this eucalyptus that's in there. Or it might decrease anxiety in women with premenstrual symptoms, or it can have other physiological functions. It might just have anti-inflammatory effects. So again, this cineol or beta-carotene or other, and they are found in a number of these plants or spices or so. So that's basically where people are looking toward to get help. So now there's a whole variety of chemicals that either have anti-inflammatory effects and or they have binding affinity for SARS-CoV-2. So here's a list of, and it's probably too small to read, and you don't need to read the individual ones, but there's a lot of compounds that have anti-inflammatory effects, and they might affect different signaling pathway, NF-kappa B, MAPK signaling, JAK-STAT signaling. So there's quite a few chemicals, phytochemicals that affect these various signaling pathways and convey anti-inflammatory effects. And then there's also a couple of compounds that have affinity for SARS-CoV-2, specifically for the a receptor binding domain, and you probably heard about that S protein on the virus. So there's quite a few. Geraniol is a prominent one, camphorol, oleanic acid. So there's quite a few compounds that actually bind to SARS-CoV-2, and that means we could potentially use those in olfactory training. So if there are these chemical components in these essential oils that we use for olfactory training, then we would think, well, they might actually help in the recovery from our loss of our chemical senses. So here, again, we're looking at clove, eucalyptus, lemon, and rose, and lo and behold, they actually do have quite a few compounds in them. Clove has, again, eugenol, eucalyptus 1,8-cineol, lemon has a couple, limonene, terpenine, citronellol, linalool, rose has, again, citronellol, geraniol, and the ones that are underlined here are known to have anti-inflammatory effects. Now, as I said on the previous slide, there are some that also have binding affinity for SARS-CoV-2, such as geraniol, which you find in here. But there's not a whole lot of them. There's, so you have to be very selective. But so what that means is then, if we find some plants that have these chemicals, these phytochemicals in them that either have anti-inflammatory effects and binding affinity for SARS-CoV-2, then we can sort of come up with our own new combination of plants or spices or herbs that we should probably take or smell or ingest because they might actually have an effect. So, if you take lavender, lemongrass, mint, and thyme, and if you look over here are the chemical constituents with anti-inflammatory effects and binding affinity for SARS-CoV-2. So you look at lavender. Lavender has binding affinity and anti-inflammatory effects. Same as lemongrass, mint here, or thyme down here. And again, they combine these two properties. And again, you have to be careful on, what's the quantity in them. But principally the idea is that in addition to the four known essential oils, people can come up with new combinations that might even be more effective in helping with the recovery from the loss of smell. So now I've talked about smell, the loss of smell and recovery from smell, and probably some of you are waiting that I talk about taste. And so what can we do to help with the loss of taste dysfunction? Well, it turns out there's actually also chemical constituents, specifically in tea. And so people thought about it and said, oh, look, so saliva plays a major role in human-to-human transmission of SARS-CoV-2, right? So if the virus is in the saliva of SARS-CoV-2 infected individuals, if we could rapidly and efficiently inactivate that virus through a beverage, specifically tea, then if you drink tea, then you actually may reduce the spread of the virus within the population. And it turns out there's actually reports that SARS-CoV-2 can be inactivated by black tea, green tea, other teas, roasted green tea and oolong tea, as well as their specific constituents here, this epigallocatechin gallate, EGCG, and some of the other components here. So tea apparently is able to essentially inactivate or kill the virus. Now, people then wondered, okay, so does tea also inactivate the virus present in saliva? And that's indeed the case. And people were wondering if it does so because saliva has all sorts of proteins and compounds in it that might affect the antivirus effect of the tea, but it turns out that tea does also inactivate the virus in the saliva, which would be a good thing. So there's a possibility that ingestion of tea may inactivate SARS-CoV-2 in saliva in infected individuals, but it remains to be determined. What's the duration and the intensity of this antiviral effect of the tea in saliva in humans. Which brings me to another point. Many foods, many things that we are eating have anti-SARS-CoV-2 effects, or they have anti-inflammatory effects. So here's sort of a graphical depiction of foods that have known or have been shown to have anti-SARS-CoV-2 phytochemicals. They're also referred to as hyperfoods. And if you can't read the names, maybe you see some of the foods here, recognize them by picture. And so these, again, may be helpful in the recovery from taste loss. So we are actually conducting a survey right now through that GCCR, where we ask people to tell us about their drinking and eating habits that might have helped them during the recovery from COVID-19 symptoms. So we are asking them all sorts of things. What are you eating? What are you drinking? What spices have you used? What essential oils? Have you used any supplements? And how did that affect the severity of your symptoms? And again, it's a worldwide survey, so we're getting input from people in different cultural areas who use different types of foods and different types of spices. And so that's basically what we are doing right now, and that's ongoing. So that's what I can tell you about recovery from loss of our chemical senses. Now this is a cover of a journal, this new journal, relatively new journal, Med, and the latest cover, it's about 10 days old, that basically talks about the emergence of new variants of concern. And it essentially says, look, there was a wave, we had one wave with one type, with one variant of the virus. And then there's another wave that might bring another variant. And again, it sort of wipes out our hopes for a post-pandemic normality. And that's true in some respect, but as far as loss of our chemical senses is concerned, it actually turns out that the Omicron variant is not as bad for our smell and taste senses as the previous ones. So people infected, so there's basically variant differences in their effect on chemosensation, and people infected with COVID-19 Omicron, with that variant, they are less likely to develop smell and taste loss compared to those infected by Delta and earlier COVID-19 variants. That's a relatively new study that came out of Virginia Commonwealth University earlier this year. They used a national database with lots of people who were tested for COVID, and then they compared the rates of smell and taste loss during the early phase of the pandemic, basically in 2020, and then with the more recent variant. And it looks like chances of smell and taste loss were just 17% for Omicron, 40%, 44 for Delta, and 50 for the Alpha variant. So basically, as the pandemic continues and new variants emerge, this is sort of good news for patients, at least as far as our chemical senses is concerned, not for everything else. And so basically what the author said is each variant has a different risk factor for associated smell and taste loss. Newer variants are likely to less likely to impact our smell and taste. But he says, okay, so initially I talked about, well, loss of smell and taste was really a good indicator of a COVID infection, right? Especially during the early months of the COVID. But the reverse is no longer true. So, I mean, it's still, if you lose your sense of smell and taste, that's still a good indicator that you might have COVID, but the reverse is no longer true. So initially, if you didn't lose your sense of smell, you might say, I probably don't have COVID. But now, do not think you're COVID negative just because your sense of smell and taste is normal, because the newer variants just might not affect your sense of smell and taste as much as the earlier variants. Anyway, so that's pretty much it for me. So I don't know if you want to go back to the question slide. And I stop sharing my slides. And if you have any questions, feel free to ask me questions.
Yes, thank you very much, Dr. Heinbockel. Please feel free to unmute if you have any questions or raise your hand. We have Zaki Sheriff.
Yes, thank you very much, Thomas. That was an excellent presentation, very informative.
Thank you.
I am working in the area of long COVID, so that's where I got a real interest in pursuing this smell and taste kind of questions. Now, you say that 45% of those who are infected with the SARS-CoV-2 virus have this smell issue, anosmia. And I'm wondering, because one of the theories about long COVID, which is the technical term for long COVID, by the way, is post-acute sequela of
Right.
SARS-CoV-2 infection. So the question is, those who have lost their olfactory sensory neurons, and you did mention that actually is replaced every, 30 to 60 days?
Right,
So,
right, yes.
Yeah, so for us, those who have these smell issues or ageusia or taste issues, especially smell issues, are the ones who are predicted to have long COVID. There were papers that came out that really confirmed that those who have this anosmia are the ones who, about a majority of those who have anosmia are the ones who may progress to long COVID. Basically, they may recover their smell issues, but they will still have other symptoms that are well documented in long COVID patients. So I'm just wondering why that would be the case. Why would the anosmia patients with COVID infection eventually, progress to long COVID? That's just one of the questions I have.
Yeah, that's a great question. I don't really have a good answer. I'm sort of have a hand waving answer, and that's sort of going back to that one slide that the patients that do not recover quickly, they typically have more symptoms, and that's basically what you just described. The people that might just have smell loss on its own, they might recover relatively quickly, but the folks, though, the patients that have several other symptoms, including loss of smell and taste, they develop into long haulers or long COVID patients. And my suggestion is that we're looking at the olfactory epithelium here, and that's good and important. Obviously, my hunch is that we need to look at other places to find out why people have long COVID. It might be, central nervous system based, or might be other parts of the body that are involved, obviously, if people have other symptoms. But I think the olfactory epithelium, I think, and also what's going on in the tongue, I think that's recovering relatively quickly. Maybe it's the nerves that are taking the information from the tongue and the olfactory epithelium and into the brain, and maybe the brain is infected as well. Again, initially people sort of said, no, there's nothing happening in the brain, and then quickly people realized, no, there actually is quite a strong effect on various parts of the brain, and that's also shown if you do MRI studies on people that recover, there's specific regions that show more activity during recovery, and other areas don't show that. So my guess is, just looking at the periphery, namely the tongue and the nose, is not really going to provide us with an explanation of what long haulers or long COVID is really doing to people.
The follow-up question, if I'm allowed, is as I said, one of the theories for the long COVID is that there is a persistence of viral presence in the tissues. I'm wondering, you did mention some RNA components of the virus may still be lingering
Right.
in the tissue or in the olfactory nerves, but you also mentioned the post-viral olfactory dysfunction. I'm just wondering if the virus is persisting, or the components of the virus, protein or RNA, are persisting. They may be, of course, eliciting immune response, and you mentioned about the inflammation that is pursued after that. But the question is, if the virus persists, usually they have to be replicating viruses, not components of the virus.
Right.
Do you have any comment on that?
Not really, honestly. You're right. I mean, I guess the question, the question is, can the components of the virus evoke the same effects or the same symptoms as the virus itself, even when the virus is not able to replicate anymore? And I don't have an answer for that, but that's, I mean, but you're right. I mean, you would expect that the virus would still replicate and should be there, but that's not really my level of analysis or expertise, so I don't really have a good answer for that.
Thank you very much.
Yeah, thank you.
We also have Nina Batia Day. I hope I pronounced that correctly. Please feel free to unmute.
Yes, I have unmuted myself. Dr. Heinbachel, you showed that the odorants that we get through the food are non-volatile kinds, and the odorants that come through the smell are volatile kinds. And then you showed at the end that certain kind of drinking tea also inactivates the virus. The third thing that you showed was Dr. Hamill's smell training test. Now, my question based on that, this information is that the pathways that are involved in processing the smell, regardless of which odorant it comes through, since Dr. Hamill is focusing, and you showed a lot of evidence about smell training through Amazon slides and everything, that is it that sense of smell in some way overpowers the smell of taste, and the pathways that are involved in olfactory sense are more intricate and elaborate, influencing the sense of taste, or it's other way around?
Yeah, that's a good question. So principally, just to go back to the beginning of your question. So when I say, actually, I think I said taste stimuli are non-volatile, because they are in our food, and they are dissolved in the saliva when we eat the food, and it stimulates the taste receptor cells. Whereas odor stimuli are volatile, but they are also dissolved eventually in fluid. Mm-hmm. The odorants that are emanating from our food, they are also volatile components. So when we eat food, there's obviously tastings in the food that are non-volatile, but there's also volatiles coming off from the food, and that's basically going through this retronasal stimulation that activates our nose. somehow, you were sort of asking, well, is the olfactory sense dominating our sense of taste? And I think in some respect you can probably say that, or certainly taste is influenced by a number of other things. That's why I mentioned, when you eat something, it might be crunchy, it might be really spicy, might have some chili pepper in it. That's not activating our taste receptor cells. That's activating our somatosensory system or our pain receptors or so. So, and then you have odorants that are coming off the food, and all of a sudden we think, oh, the food smells good. I'm not really tasting it. But so in that sense, you could say, well, taste is a lot more than just taste, than sensing tastings in the food. And therefore, you could say, well, maybe then the other senses are overshadowing what we are actually tasting. That's certain. I guess you could make that argument. So I'm not so sure if that addresses your question.
To some extent it does. Thank you so much. The idea was that since sense of smell, it's more wide and that is being enhanced, perhaps it is more, what should I say? It is probably, sense of smell is more vulnerable to the viral infection in comparison to sense of taste. Is that accurate?
Yeah, that might actually be true too, because I think probably more people have loss of sense of smell, and the loss of the sense of taste is recovering quicker than the sense of smell. So in that sense, you could make an argument that smell is affected more strongly by COVID-19 compared.
Right, that's what it seems like from the results. That's what I was concerned about.
Right, yeah.
Thank you.
Yeah, thank you.
Dr. Joseph Hannig has a question. Would you like to share, Dr. Hannig?
Yeah, so the question I had is, are the effects of tea on the virus just in saliva, or could it have beneficial effects systemically? And has anybody speculated about what the active components are that might be beneficial?
Yeah, I think as far as the active components are, I think that ECGC, that gallate component, I think that's one of the compounds that has been tested specifically, several of these compounds. But I agree. So there might be a systemic component as well. I'm not so sure people have addressed that yet. I'm not aware of. I mean, these are papers that came out not too long ago, last year. I mean, everything's relatively new. There might have been some follow-up studies that actually looked at systemic effects of drinking tea, and whether, I mean, that's probably, if you talk to tea drinkers, they would probably say, well, tea drinking is not just, good in the mouth. It has, generally beneficial effects. And so you could make the point, but I'm not immediately aware of studies that have addressed that question.
Thanks very much.
Yeah, thank you.
Are there other questions from the audience? Thank you very much, Dr. Heinbockel, for taking the time to tell us about your research. It has been really, really fascinating. And if anyone else has remaining questions, please feel free to email us at rcmi-cc@howard.edu. I placed it in the chat. It's also up here on the screen. Thank you very much for joining us for this journal club session. Please feel free to join us for our next journal club. It will be on October 24th, entitled The Problem of Coagulation in COVID-19, featuring Dr. Brukatachu. I will be sure to send a follow-up email with the registration link, and we would love for all of us to join. So we will see you on the next Journal Club. Take care and have a great day, everyone.